Thank you. Thank you very much for the introduction, and thanks for the invitation for this opportunity. So the title of my talk is Topological and Geometrical Approach in Modeling Spatial Memory. And then basically I'm going to go over some basic ideas that we're using in trying to connect neural activity and cognitive phenomena. So the big idea, the big question that I'm going to address in this talk is essentially, how does brain represent space? Now, this question actually is interesting not only from the point of view of neuroscience, but also from the point of view of physics and maybe mathematics to some extent, because if you think about it, what do we have? Like, what does brain do? It's a large collection of neurons, spiking neurons, and they're producing very complex neuronal codes. So just complex temporal codes. And the question is, what is the link between temporal codes on the one hand and the phenomenon of geometry on the other hand? It's clear that the brain in some way is capable of integrating this vast amount of information and produce this geometrical realm. And how do we actually address the question where the connection comes from? That's the main question I would like to address. Now, of course, the data that we're using for this kind of analysis is never coming from humans. Unfortunately, we don't have access to human data yet, but we're using data coming from rodents and from primates mostly. And in analysis, in our attempts to understand how rodents represents rats primarily, I'm going to be talking about rats, how rats represent space. The key concept is the concept of cognitive map, by which we understand, as it's written here, it's internalized representation of the environment that enables spatial cognition. Now, the key word here is the word internalized. And what do we mean by this is that spatial representation, and it's not a reaction of the brain to sensory inputs. Sensory representation, spatial representations emerges from endogenous dynamics and neural activity of complex nexus of networks in different parts of the brain. And the question is, how does this how does this happen? How does the brain collect data from different parts of the brain, spiking outputs, and integrate them in some sense to produce a coherent representation of space as a whole? Now, in order to begin to address this question, we need we built our modeling on empirical data. The way it works is that we record data. At this point, probably I'm saying some very, very basic facts, but I'm just going to go over this nevertheless to make it more coherent, my story. So we basically record data. Coming from here, we have this simple schematic representation of tetras going into neuronal tissue. You have this up to scale representation of tetras going into neuronal tissue, and we collect spiking data. And the idea is to, in that way, we were able to identify, a bunch of what is called spatial neurons, whose spiking activity correlates with geometry and topology of space. And the key example of spatial neurons is, of course, hippocampal place cells. Everybody knows them. They're famous for, highly selective firing activity. Every place cell is firing in a small domain in navigated environment, which is called place field. So every place cell has a place field, and together these place fields, they form what is called a place field map. When I'm saying the word map here, you should understand it in the same sense as a geographical map. Basically, it's a map of locations. And another example of spatial cells are head direction cells that fire when the animal's head is oriented towards a particular direction in space. Another famous example is grid cells, which I definitely won't have time to discuss today, but it's cells that are firing at the vertices of triangular lattice in a space. And this is the level of empirical information that we actually are getting from the experiments. And the question is, okay, how can we process this type of information that is conveyed to us by this spiking populations of neurons? And specifically, there are two big questions that I would like to address. Is number one is, how does the brain interpret spiking data intrinsically, because if you think about this, I keep mentioning those place fields and head direction fields and grid fields. And if you think about it, the brain doesn't really have access to this kind of information. This is how we, as experimentalists or as theorists, this is how we, visualize our data. But the brain, what the brain has access to is just, spike trains, temporal codes. And how is it possible to interpret one type of, spiking activity as a representation of locations, as opposed to, another pattern of locations is representing orientations? And the next question that comes after that is how the brain is capable of combining different types of information to produce a coherent representation of the environment as a whole. This type of questions have, as I mentioned before, it's not only neuroscience, but to some extent, physics and mathematics are going to be involved. Now, to address questions like this, first thing that I would need to do is, and I should say that from now on and probably until the very end of the talk, I'm going to be talking about place fields and place field map. So to understand the structure of representation that is produced by hippocampal place cells, first thing that I would need to do, I would need to formalize information that's contained in a place field map. And the way I'm going to do this, it's a very natural, straightforward approach. Every place field that I find in the place field map, I'm going to represent by a vertex. If I have two place fields overlapping like this, then that is going to be represented by a link. 3 place fields, it's a triangle. 4 place fields is tetrahedron, and so on. You can these objects are, what I'm describing here are fundamental objects in algebraical topology, and they're called simplexes. And basically, when I'm saying the word simplex, I mean what is called an abstract simplex. Basically, it's a list of elements. In this case, it's a list of place fields that are overlapping in space. But it's very convenient to think of these simplexes as of polytopes that are represented by this, the sequence of shapes, a point, a connection, a straight interval, triangle, and so on. Now, it's pretty easy to see that you can actually, any configuration of place cells can be represented by a configuration of simplexes. It's one-to-one correspondence. And specifically, if you have a place field map, the entire place field map is represented by a combination of simplexes like this. And this combination is, in algebraic topology, it's known as simplicial complex. I'm hoping that people, I probably have heard of the concept of simplicial complex. And this specific kind of simplicial complex is called the nerve of the cover. So the cover is the place field map, and the nerve of the cover is the simplicial complex that we built in this way. Now, the key idea that connects, that underlies this approach is a theorem proven in 1930s. It's by Alexandrov and Čech, who actually were able to argue that the shape of the simplicial complex is the same as the shape of the underlying environment. There are some requirements are needed to make the statement precise, and I'm going to go into more details as I go, but basically you can think that the simplicial complex, the nerve of the cover, has the same number of pieces, has the same number of holes, the same overall topological shape as the underlying environment. So this is kind of the first implication, the first argument that we can give to say that place field map, it knows something about space in which the animal navigates. It's again, I'm going to always make this connection between physiological statement and kind of theoretical modeling statements. It's a common thing to say that, place cells represent space. And starting from this point, I'm going to ask question like, in what sense place fields represent space? Like, how do we deduce information about space in which we navigate from spiking activity? And this theorem is the first hint that if I collect information that's contained in the place field map, I can actually deduce the topological structure of my space by building what is called this nerve complex. Now, the issue that if we consider more closely what I just said, is that every element of the cover is fundamentally, it's something that was constructed using spikes right? Every place field, it's a certain representation of spikes that I'm using to kind of visualize my data. But so in a sense, this is one way of approaching that question. How do I integrate spiking information to be able to say something about my space or to construct a space eventually? Now, one big idea here is that, as I mentioned, several times already, is that those place fields are simply visualization of, information contained in spikes. The brain itself may not be using it. It's something that we're using to kind of help our thinking process, basically. In reality, what brain has is just a bunch of spikes. And the question is, how do I make this connection? As you see here, I don't know if you can see my cursor here. Yeah, it's like, how can I go from here to here? How can I integrate information and go directly from spiking activity to constructing an object such as, this nerve complex using only temporal arguments? So basically what I need is to produce this kind of construction only using temporal relationships produced by a population of neurons. And in fact, this kind of construction is easy to do, because if you think about this, imagine if we are observing a spiking activity of a cell. What it means is that at that moment, the rat is passing through a place field, essentially, right? And we agreed that we're representing a place field by a vertex. So now I can basically think of either this or this temporal representation or spatial representation. I can think of each one of them as, I can represent each one of them by a simplex. Now, if I have coactivity of two cells are being coactive at the same time. What does it mean? Is that at that time, the rat is running through an overlap of place fields, and I can think of every instance of coactivity of two neurons as a representation of overlap of two regions. In principle, I can think about, place fields, or I can think about representation of regions in more abstract sense, and represent that with overlap of, one-dimensional simplex, a link between two vertices. Same thing with overlap of 3 cells, 4 cells, and so on. So what is happening is basically I have this one-to-one correspondence again between pattern of coactivity of neurons and overlaps of, of regions, and I can carry on my construction. And let's say here I'm showing a schematic representation of 7 spiking neurons. And what can I do? Like, first of all, every single neuron is going to correspond to a vertex. Every time I have an overlap, a coactivity of 2 neurons, that's a link between the corresponding vertices, right? I can look at all of them. And in that way, if I have triple coactivity, 3 cells are active at the same time, that's a triangle and so on. And that way, it's a direct analog, a temporal analog. And basically what I've done is I'm building a temporal analog of nerve complex, right? And the idea is that I'm going to be able to use this construction to extract topological information about space in which the animal navigates. Now, this is all fine, but I would like to mention here, there's actually a very important difference between construction of the nerve complex as it's represented in in standard algebraic topology textbooks and this construction. The difference is that spike trains, if you think about them, spike trains are unfolding in time. The neurons are producing information over time, right? Which means that if I'm building my simplicial complex, it also is going to develop in time. Its topological structure is not going to appear at once. Is going to develop slowly. And basically what I'm going to be able to do, or should be able to do, we should look at accumulation of spike information and translate it into how topological information is being produced. And from physiological perspective, basically we can interpret this process. One way of looking at this is to look at this process and say that, it may be representing learning, for example. How do we extract qualitative information about space? Slowly, by observing how information unfolds, how it's provided by a population of neurons over time. So in that way, for example, I can actually follow how ensemble of place cells is slowly producing that cognitive map that hippocampus is believed to produce. Now, the question is, this is basically an idea. And the question is, will it actually work or not? Because we're actually making quite a jump here. We're starting from relatively standard and classical concepts in topology, but we're kind of translating them into temporal domain. And whether will it work or not work, it's a question. Now, before I go into discussing this question, I would like to make a short kind of, 5 minutes just define mathematical terms here. When I'm saying our goal is to describe topological shape of that simplicial complex. Now the question is, what does it mean to describe a topological shape? Like, what does it take to describe a topological shape of a cup, for example? In principle, there are many ways of approaching this question, because there are different languages, different semantics that we can use to define what a shape is. A very concise and succinct way to do this is to describe a shape in terms of, how many loops it contains. Now, when I'm saying the word loops, you probably think of, one-dimensional circle like this. There are natural generalizations of one-dimensional loops. You can think of zero-dimensional point as a zero-dimensional loop. A sphere, it's a two-dimensional surface with loops on itself, so it's a two-dimensional loop. And so on, like in a three-dimensional sphere with a three-dimensional loop and so on. The question is, how many loops can I find on a given shape? Like on this cup, I can find a point, that's my zero-dimensional loop, or I can draw a loop here, or I can draw a loop here. And, question is basically to list possible loops. Now, next thing that I have to ask is, how am I going to count loops? Okay, I just have to define how do I count loops, basically. So the idea is that if you can take a loop, one loop, and deform it continuously into another loop, it's the same loop. So we count loops up to continuous deformation. So let's say this zero-dimensional loop here, if I can shift it over and move it over here, is the same loop, only shifted. Similarly, if I take a one-dimensional loop here and I can make it bigger, it's the same loop. Right? And if I can make it smaller, it's also the same loop. If I can contract a loop into a point, it's actually not a one-dimensional, it's not a one-dimensional, it's a zero-dimensional loop. So everything is counted up to these smooth topological deformations. Now, in case of this cup, this loop, for example, I can contract this loop, but not this one. So the fact of having this loop is telling me something specific about the shape of my manifold, this cup in this case. And same thing in 2 dimensions also. I can take, as long as I can take any loop in any dimension, as long as I can deform it from shape A to shape B, is the same loop. Then this is this infamous example that connects a coffee cup to a donut. I apologize for using it, but it's a good illustration. So the list of loops that I can identify on a given shape it's a very crude characteristics of a shape, but on the other hand, it's enough for us to identify the kind of shapes that we're going to deal with. It's a very crude description of a space, of a manifold or simplicial complex, just a list of loops in different dimensions. So in this case of just to illustrate this, on this cup, I can draw 2 loops I cannot contract. And if you compare it to this list, is the same list of loops as in this case. So by listing loops in over non-contractable loops in the cup, I can identify that this cup actually has the shape of a torus. So this is the kind of language that I'm going to use. And it's crude. Like, for example, this cup and this cup are not going to be distinguishable in this language, but for more general variety of shapes, it's enough for our purposes. We'll be able to say how many holes a given shape has, how many sheets it consists of, and this is enough for the kind of analysis we want to do. And basically, we'll have to apply this language to understand how simplicial complexes are organized. Now, next important idea is that, oops, what am I doing? Yeah. So next important idea is, if you remember this slide that I used to define what is a simplex. Simplex was related to overlaps between regions in space. But now, if you think about this work, our goal is to understand how things develop in time. And those place fields in this specific case are, we don't necessarily need to think of them as of static objects. Imagine that as information is being accumulated, the regions that we're using to construct simplexes or other like instances of information, they can actually change structure. They can, like in this case, regions can change sizes or shapes, and that would affect the pattern of overlaps, and that would affect the structure, the simplexes that are contributing to simplicial complex. And the question is, if I allow the elements of my structure to change in time, how topological shapes are going to develop in time? This is basically to say that standard topological language that people are using in algebraic topology to define simplicial complexes may not be enough. And the problem is that in classical description of topological complexes is too static. We need kind of a temporal component. So imagine if I would say have vertices that represent, you can think of, for example, centers of place fields. And if they would do something like this, they would change shapes. How the topological information that is encoded by overlaps is going to develop over time. It's a conceptual question, how to address dynamics of topological information. So here's one example how things can work. So what you are seeing here is, imagine I have these vertices, and these vertices, imagine you have vicinities surrounding every vertex. And imagine that every vicinity is changing its size. As you know, I probably have to play it again, but basically what happens is that when vicinities are small, so over here, it's not, okay, so this axis, let me actually show, I have still pictures, easy to explain. So imagine what you are seeing here, this axis represents size of the vicinity of every vertex. This is just, as that size is changing, you start, maybe I should start from here again. Let me play it again. I'm trying to, okay, imagine, so what is happening, like every vertex here is surrounded by vicinity. Vicinity is growing in size. And as vicinity becomes bigger and bigger, you begin to establish more and more connections. And these connections may form loops. They may form loops in 1D, in 0D, on 0D just vertices, or in 2D. We're only following like in dimensions 1, 2, 0, 1, and 2 in this case. So if you see, now this is kind of illustration of what is happening, but now let's look more closely. So I'm hoping that now you have this picture in your mind that when size of the vicinity is increasing, right? What you have at the beginning, when vicinities are small, you have a bunch of vertices. This is the list of zero-dimensional vertices. You have many of them. But then the size of the vicinity, when the vicinity grows, part of these vertices begin to disappear because they begin to produce links, and you start having loops here. And this second block of this second diagram, it basically represents loops that appear in 1D. They appear as the size of the vicinity is changing in this range. But then vicinities grow even bigger, and you start having two-dimensional loops. So you see this kind of dynamics, what is happening as the size of vicinity is changing. And one thing to notice here is that we're actually having quite a few loops we are seeing on these diagrams, but most of them are transient. They appear at a certain moment and then they disappear at a certain time. So all these zero-dimensional loops, they exist over here and then begin to disappear and they go away. These one-dimensional loops, they appear and they go away. However, there is one loop that's at the very bottom that appears and it never goes away over the entire process. These one-dimensional loops, all of them are transient, but this two-dimensional loop, it starts here and continues forever. So basically what we are seeing here, that some of this, like how would I say it? Some of the loops are persistent. They persist, they're stable. In simple terms, they're simply stable. And because they're stable, we can make an assumption that these stable loops represent physical, physical information about our system. And in this case, if you look at what is happening, we went through this transient period of loops appearing and disappearing. At the very end, we have one zero-dimensional loop, nothing in 1D, one two-dimensional loop. And the question is, okay, this is a topological barcode. What does it represent? And if you think about this, the only choice that you can come up with, the only interpretation of this information is just that this is a sphere. It's a one zero-dimensional loop. Basically, I have one point that can move everywhere. So which means that we have one piece. Your space consists of one piece. It doesn't have one-dimensional loop because it's contractible, and it's two-dimensional because it has one two-dimensional loop. So the only thing that can actually satisfy all these conditions is a sphere. So this kind of logic we're going to apply. And another example, this is, if I play the same game with a torus, right? Again, I pass through the period of vertices, and then these vertices produce transient one-dimensional loops. And then I start to produce a two-dimensional loop. And in the end, I realized I have this topological barcode, one piece in 0D, two loops, non-contractible, and it's a two-dimensional thing. The only choice is it's a torus. So this kind of example, well, the same thing in 1D. I don't have to go into more details. And now, if you remember this slide, I used it 5 minutes ago. That was showing that, as spikes are produced by a group of neurons, you have coactivity complexes, this topological, temporal analogs of nerve complex. It also develops in time. And now my goal is to follow how this topological information is forming, or how is it being produced from the point of view of persistence analysis. I'm going to compute loops, and I'm going to see which loops are persistent. Like in this case, these two loops, right? So if I look at this, by the way, it's one loop, and different colors correspond to different dimensions in this case. So nothing in 2D, one-dimensional loop, and one zero-dimensional loop, so it's a topological circle. So this kind of analysis, I can apply this time now in temporal domain. So it's the same game. I'm starting from spatial configurations. I'm using them as a motivation, and then translating that picture into temporal domain to see what's going to happen. And by the way, again, notice that everything is developing in time now. So I can, for example, ask questions like how much time it took to produce stable topological information. And if I'm thinking of what neurons are doing, they are producing this topological representation of space, I can interpret the time that is necessary to produce stable topological information as learning time. It's kind of, to facilitate interpretation of what is happening. Now, and the idea is, of course, that, this now, if I'm going to apply this to describe, the information dynamics, information that is produced by place cells, I would imagine that this persistent loops, this information should capture the shape of space. And once again, the key word here is the word should. But will it or will not? It's a question. Because, like, if you think about it, if this approach has any meaning, if it applies to biological systems, then we can right away say two things. Number one, this information should be actually explicitly extractable from spiking activity. And to some extent, we answered that question because we explained how can I actually build simplicial complex. So basically, this is constructive part. And, we, in principle, can argue that, yes, we can more or less extract this information, that we can build simplicial complexes. But second issue is that topological shape or topological information that is contained in simplicial complexes should be reliable. It should be reliable despite several natural sources of randomness and stochasticity and variability, like neuronal spiking is stochastic by its nature. Animal behavior. Imagine like how these place cells are going to fire in very random orders, and nevertheless, the information that is produced by spiking activity should be stable. Or you can imagine that the animal is running in spaces of very different shapes. And every time this approach has to produce reliable results. Will it work or not? You can imagine that, if your spiking activity is reliable, if your, spikes are produced reliably, have nice, fat, good-looking place cells or place fields, then maybe yes, things might work. But if the information is not sufficient, then maybe not, right? So at this point, it's clear that the whole idea of applying topology to spike analysis, it definitely would depend on parameters of neuronal activity. In some cases, it may work, in other cases, may not. Okay, question is, how do we check for which parameters things are going to work? Now, the minimal set of parameters that we would need to use is, for example, we can characterize every neuron by its firing rate, for example, by its maximal firing rate, and by place field size. Now, this is immediately a problem because imagine if we're talking about in hippocampus, there are thousands of cells active in small environments. So we immediately talk about several thousands of these parameters. But what we can do is we can look at the statistics of these parameters. And in that case, we can think of every individual firing rate as an instance of a random variable. What is important is not the actual value of F, of firing rate, or S, the place field size. What is important is the shape of the distribution. And every distribution is going to be characterized by, let's say, by the mean or the mode of the distribution. And these numbers are just instances of this random variable. So with that approach, this kind of statistical approach, I'm going to characterize my entire ensemble of place cells by just three numbers. It's going to be the mean firing rate per ensemble, mean place field size or typical place field size, and the number of cells. So with this shift, we're now talking about just three parameters, three-dimensional parameter space, which is kind of manageable. And so, yeah, every ensemble of neurons is going to correspond to a point in this three-dimensional parameter space. And what I can do is I can basically test different, place cell ensembles by moving in this, parameter space. And the kind of a game that I can play is the following. I can, for example, imagine we take an environment, like this simple environment, like the rat is running, it's 1 meter by 1 meter. We usually simulate because this is what people are using in experiments. It's a very common size and shape of the environment. And now what I can do is I can simulate place cell activity in this environment and try to build my wonderful temporal simplicial complexes and see what kind of shapes do these complexes assume for different parameters. And since I know what kind of a space I'm dealing with, right? I know its topological shape. I know its barcode. It has to be 1 in 0D because it's one piece. It has to have a hole, and there should be no loops in higher dimensions. So I should be able to get if place fields, if place cells are operating properly, then I should be able to see this answer. And the question is for which place cell ensembles this answer will be produced. Now, if we run the computation, what turns out, actually, this is the answer, the first level of answer that we're getting is that number one is that not every place cell ensemble is capable of producing the correct answer. The correct answers are coming from a domain in parameter space, which we call learning region, in which, basically this learning region restricts functional ranges of spiking parameters to a certain domain. And by the way, and what's interesting is that, what I've said so far, I basically, was referring to physiology as to, basically physiology was only motivating my questions. The questions themselves were essentially purely theoretical. We said that we're going to build a simplicial complex. The simplicial complex may, represent space correctly or incorrectly. And then we run this computation and, even the fact that we got some domain in parameter space, that domain could have laid anywhere in parameter space. For example, in order to produce a correct topological map, your place cells are required to fire with, firing rate 500 hertz, for example. It could have happened like that way, but it doesn't. What's interesting is that the domain of regions, so the domain of parameters that are required to produce correct topological space, topological shape of simplicial complex, they more or less coincide with physiological domains. So this match is important because it's telling us that this approach has something to do with reality. Okay, so and if you look at this, first of all, I should say that the colors here represent learning time. If you remember how much time is required to produce topological maps, so this is a key parameter that we're using. And when we run our computations, we study each map, we simulate each map some 10 or 12 times, and then average the results. So every point here has a color, and color corresponds to the mean firing rate, mean learning time. And what you see is happening is that longer learning times are sitting at the periphery, and shorter learning times are in the core. And if you are getting outside of learning regions, even though your place fields are seemingly firing, place cells, are seemingly firing in kind of, place-specific manner, nevertheless, they are not somehow, like, information is not being processed properly, and it's difficult to explain it intuitively, but the computation shows that, if you are falling outside of learning region, even though seemingly place cells behave like, there's nothing too special about what kind of place field they produce or, any other obvious parameters like this, you are not able to produce correct topological maps. So you have to sit here in terms of parameters that you are using. Now, an interesting thing is that if you, like we tried, using several physiological recorded data, we didn't have access to too many cases, but if you actually look at the distribution obtained from actual recordings, that these are histograms for firing rates and place field sizes obtained from a rat actually navigating an environment. And you see that, this specific ensemble falls somewhere inside that the learning region domain. So there is this nice correspondence. And we did also several other trials, and in some cases, those trials were purely computational. In some other cases, we had some data, but I think that is going to be illustrative. I'm going to bring several examples to kind of give you intuition how this would work. So first case, ethanol. Imagine the people have carried experiments in which rats were given distributed alcohol, and what happens when the rat gets drunk is that place fields are getting larger in size. It's a very common kind of observation that people see in rats when distributing alcohol. Now, in terms of our model, what it means is that place cell ensembles were sitting somewhere inside the learning region because of this phenomenon when place fields are becoming larger and larger. What happens is that that ensemble of place cells is drifting outside of learning region. So you may acquire, it's of course it would require more data and more analysis, but a possible scenario might be that the ensemble is basically getting outside of learning region, and it's a common knowledge that people also complain that they don't remember things after getting drunk. But the thing is that they are not. It's not that they cannot remember. The problem is that they did not form memories in the first place. And this analysis can actually show why the memories would not show, because the topological map cannot form, the correct topological map cannot form. Another example is the case of Alzheimer's disease. In Alzheimer's disease, what is happening is that two things are essentially happening. Place field sizes are getting larger, and number of cells that are active is dropping. And certain things are happening to firing rates, but these are the key phenomena. And if from the point of view of this model is that you are actually, when this happens, it's as if you are trying to escape from learning region along two directions. You are getting outside along place field size, and you're reducing number of cells. So it's in two ways you're getting outside of the learning region. Now, case 3, aging. In aging, similar things are happening. Place field size are larger, getting larger and so on. But another interesting phenomenon that happens is that place fields become patchy. What I've said so far is that every cell is firing at a particular region, but this only, it's not literally true. Like there are many cases in which a given place cell can have several like disconnected parts of a place field. And when this happens, you still can run this argument, although Alexander Čech's theorem would not work literally, but still you can carry out this construction and ask the question whether the topological shape of the resulting complex is going to be correct or not. And this patchiness of place fields usually easily destroys integrity of your topological information. The shape of topological shape of your simplicial complex is deteriorating, and physiological, this is what happens in aging. It's one way of understanding why like memories are, it's more difficult to form memories for aged subjects. Now, in learning, what is happening, an opposite process. Now, what they are saying here, this is a circular environment like this, and this hotspot represents a place field. So what is happening as time goes, place fields becomes more, place cell activity becomes more sharply tuned. Your place field becomes smaller and more concentrated in specific domains of space. And what is that in terms of this model? What is happening actually? It's opposite process. You are moving from outside, from periphery of the learning region into its core. So, and also one thing I wanted to mention is that the shape of shape of the learning region actually depends on the shape of the environment. If your environment, let's say, is a simple linear environment, then the topological map will pretty much work for almost arbitrary parameters. But if you are thinking about either complex two-dimensional environments, or let's say in case of bats also have place fields, but these place fields exist in three-dimensional space. And in order to, build a reliable map of a three-dimensional space or complex two-dimensional space, you have to, impose more restrictions on the firing parameters. And as a result, the learning region becomes actually smaller. So the shape of the learning region reflects complexity, I would say. It reflects complexity of underlying environment. Now, with this model, I would like to take it down several steps. We can actually study the effect of a set of different physiological parameters and physiological phenomena. For example, what I mentioned before was that place cells fire in specific domains in space, in the place fields. Now, this is not the entire story, because the place fields actually, their spiking activity is coupled to what is called brain waves. Now, brain waves is basically the result of the phenomenon of synchronization. Like large ensembles of neurons in the brain, they tend to synchronize and produce this oscillatory mean electromagnetic field in coupled across different parts of the brain. LFP, local field potentials. And place cell activity, for example, is coupled to, strongly coupled to what is called theta wave. It's an oscillatory process that on average has the frequency of about 8 Hertz. And when the rat is running through a place field, for example, its spiking happens at the preferred phases of theta. Like, see what is happening, right? Like the theta wave continues to have about, I would say, maybe 8 or so periods of theta across a place field. And when the rat is running through the place field, it spikes in, discrete places that are associated with specific phases of theta wave. It's a kind of complicated process. And it's commonly believed, and there are many experiments that in different ways argue that place field activity, that theta modulation of spiking activity is important for learning. Now, in the model, we can actually test this idea in a very direct way, because it's a computational model, so we can actually numerically introduce this coupling between phase of theta and spiking, and then can turn it off and see what's going to be the effect. So what you are seeing here is the case that this is the learning region in a rat when the rat is running in this environment, and there is a coupling between theta wave and spiking. But if you remove it, this is what happens. It basically means that a bunch of ensembles that in principle were capable of producing reliable maps of space are losing this ability. And if you go, and this is basically a direct illustration of the importance of theta modulation for successful learning of space. And you can go into details, and you can just basically estimate by that learning times are increasing by over 50%, and statistical stability is going down. And if you look into the structure of this topological mapping, we have all of a sudden we have this large number of topological defects that won't go away. Like in terms of these loops, you will have the spurious loops that would start and they're supposed to go away at some point, but they don't. They persist, and basically your map loses quality. And this is an illustration that theta wave actually allows to remove all that by basically introducing extra synchronization between spiking activity. And an example, actually, this has a direct, this kind of analysis has direct analogy, physiological analogy. As it turns out, like the way to suppress, it is possible to suppress, experimentally suppress coupling between spiking activity and theta wave, and that is to distribute cannabis or marijuana to rats. As you would imagine that, if you distribute marijuana to rats, of course, their learning ability goes down. But what's interesting, if you look at the spiking activity of neurons, it turns out that the maximal firing rate doesn't change so much, and place field sizes don't change so much, but the coupling with brain waves is disturbed, it's destroyed, basically. And if we, like from the point of view of the model, this is what happens. If the control mice, or rats, we were looking at rats actually, have large and well-defined learning regions, and then, learning regions are shrinking, meaning that, we don't know where in this domain that a specific rat is, but statistically speaking, what is happening is that many place cell ensembles were capable of representing space with theta precession, lose this ability, and the rat is not capable of performing as well. An interesting outcome of this model was that we applied it to the case of bats. As I mentioned, the bats are representing or mapping three-dimensional spaces. And what's interesting, they observed experimentally, there's a very little theta precession. So rats actually, they do have theta waves, but the coupling, I believe the coupling between theta wave and spiking activity was suppressed. So we looked at what is happening, modeled this case with this topological model, and what we saw is that this is the environment that we modeled, and this is a list of loops, like I was explaining before, like 0-dimensional, 1-dimensional, 2-dimensional loops. This is when theta precession is on. This is when theta is off. And surprisingly, when theta is actually coupled with spiking activity, the topological structure of the map, this simplicial complex, is actually, it's riddled with topological defects in 1D and in 2D. But if you turn theta off, then all these topological defects disappear, which is to say that, theta modulation in bats somehow amplifies topological noise. And it's, we can speculate that how to explain why this happens, and my current understanding is that basically when in 2D you have you produce a lot of spikes, and when you synchronize spiking activity with theta wave, synchronization of spiking activity, it helps to build a topological map. But in 3D, neurons are place fields are not visited as often because you have too much space, literally. And when you're introducing an extra condition, extra constraint on your system, that constraint by itself, it worsens the process of of these topological computations. And somehow, like in the end, if you just do the computations, the modeling, it turns out that theta in 3D, it precludes from building efficient topological maps. In 2D, it helps. I wish would, we actually found this result after this effect was observed experimentally. I wish we would be able to, do this kind of observation before they observed it in the experiment. That would be a beautiful prediction of the theory. But... Okay, so several more theoretical questions that we can address with this model. The way I was describing this whole procedure, we were basically looking at spikes and coactivity of spikes. We're interpreting as this overlap of regions and so on, right? Now, when I'm saying, when I talk about coactivity of cells, what is coactivity? Is that I'm observing spike activity of one cell and another cell over a certain period, like period of coactivity, W, right? You can imagine that if this W is actually changing, the entire picture is going to change very significantly. If W is too small, basically you require two cells to be coactive over a small period of time, then such coactivities are a bit difficult to detect, and vice versa. If W is too large, you may detect wrong coactivities, for example. So the question is how things would depend, but with this model, what we can do is we can treat this W as a parameter. And just basically investigate what will happen as W is changing. Like just a purely theoretical question, like how much W would be, how things would depend on this W. And this is how they basically depend. What you are saying here is, this axis shows the size of temporal window that defines coactivity. And this is learning time. And so what you see is that, first of all, when coactivity, when W is too small, then of course you stop, you cannot encode topological mass because W is too small. You basically don't detect enough coactivities. That's easy to accept. Another interesting thing is that W cannot be too large, because it's also in principle easy to understand, because if W is too large, then any cell is coactive with any other cell. And as a result, what you get is just a big gunk of space with no structure, and it's a wrong answer. But in the middle, we have this interesting inverse hyperbolic dependence, and should be proportional. So basically, learning time is W to power like 1.1, 1.2. And by the way, I don't insist on any specific numerical value. What is important here is the qualitative behavior. And the qualitative behavior is that you kind of have this hyperbolic curve. Now, so this would be a kind of a naive prediction of the theory. However, we can do better than this, because if you think about this, look at this domain. What is happening here when W is changing over here? The learning time depends very strongly on W. So basically, you can think of this as an instability. Of course, like in the real physiological system, this W can actually, you would imagine that, it can change, first of all, from neuron to neuron, and in general, like W can actually change. But we would expect that if the system is stable, then the learning time should not react as violently to change of W. So this kind of left end of this curve is not good. And the same thing about this part, because what is happening is that it's purely numerical result. Is that if you try, you look at the statistics of correct outcomes, the circles, I forgot to say, the circles are telling you what is the percentage of correct outcomes in simulating different topological maps for different Ws. And you see that this percentage drops down. So basically what we get is this domain over here is what would theory basically predict. Here's where W should live. And, amazingly enough, it actually does correspond more or less to the coactivity window that is defined in the experiments. And before we did this study, we used about this size of W for our previous computations, but then eventually we're able to essentially derive this. I don't know if it counts as derivation, but it's basically it's a numerical experiment, but it leads to this kind of a conclusion, which is matching the experiments. What else? Another, what I've described, theta modulation of spiking activity, right? But in the brain, there are several different kinds of waves, and another very important kind of a brain wave is what's called gamma wave, which has frequency about 7, 8, 9 times higher than theta wave. And what's interesting is that what I've mentioned before that when rat, I'll try to explain this slide the best I can, is that spikes were, before I said that spikes are produced at a particular phases of theta, like here, 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 right? But in reality, what is happening, that every preferred phase of spiking, it corresponds to a particular period of the gamma wave. So frequency of gamma is about, 8, 9 times higher than frequency of theta. So I have about 8 or 9 gamma periods over each theta period. And when the spikes are spiking, like spikes are being produced, they're being produced within a particular gamma period, like this. So it's like, imagine this is gamma over here, and the spikes are kind of corresponding to a particular period. So to describe the effect of gamma wave, what we need to do is to understand how gamma waves are coupled to spiking. So this would be a loose coupling, and this is tight coupling, something like this. We need to basically find a way to do this. And a simple association that we can have in this connection is that, okay, if these spikes, if we would think of them as of particles that are sitting over a potential well, U of x, think of it as a potential, right? And these are particles for a second. Then, this potential will have some width and will have some depth, and the depth corresponds to amplitude of the gamma wave, right? So it's, again, it should be proportionality sign here. So you can think by, invoking arguments in the level of maximal entropy principle, we'll say, okay, the probability of spiking, you can introduce this extra Boltzmann factor, that the probability of spiking is modulated by e to the power negative, like the depth of your potential, and you would introduce an extra variable parameter, effective temperature. Of that effective temperature will basically describe how tightly those spikes are sitting around a given gamma period. And basically now what we can do is we can introduce this gamma modulation and talk about hot or cold simplicial complexes, depending how big or how small this beta is. I don't know what is happening with this signs, but basically if beta is close to 0, it meaning your effective temperature is high. Spikes are spread very widely around, gamma periods. That would correspond to a hot, in this terminology, it's kind of a funny terminology, but, it's kind of suggested by the logic of what is happening, that this would be a hot simplicial complex. And if simplicial complex gets cold, that the effective temperature goes down. Yeah? Then the spikes are being concentrated at the trough of gamma. And if you run the simulation, this is what's happening, is when you have high beta, I'm sorry, that something is wrong here, is if beta is small, for small beta, high effective temperatures, you have large number of topological loops, large number of topological defects. I'm showing here is 1D, but in 2D happens the same thing, and large learning times. But if the effective temperature goes down, in other words, spikes are concentrating at the gamma troughs, then learning time goes down also, and these topological defects, it's as if when you are lowering this effective temperature, you are kind of freezing out topological defects from that simplicial complex. And should also say that the physiological value of this effective temperature, or beta inverse temperature, is around 2. So we can actually follow how this happens, although we did not have good enough data to actually run it, to apply it to data analysis, but this is something that in principle, I guess, can be done. So this is what happens. Again, just one more slide showing that when you go from hot to cold, here's what happens. You have a lot of loops that are slowly disappearing, and you eventually get correct topological signature. And you see that when temperature is high, you have only few 1, 2, 3 ensembles. What we are saying here, this line, these are cross sections of learning region, and you can see that what is happening, that when temperature goes down, you have more and more and more place-cell ensembles that are capable of representing correct topological properties of the environment. So yeah, and we know it's a well-known fact that gamma modulation is very important for learning, and this approach gives another perspective of why this should be happening. Now, one more thing that we can take, one more perspective we can address here is that when we built our coactivity complex, basically what we're saying that every instance of coactivity, several cells, like in this case, like 4 cells are coactive, they correspond to a simplex. Simplex contributes to the simplicial complex, and I'm talking about its topological properties. But in reality, it's important that when the cells, place cells in this case, are producing spiking activity, but what's important is that the spiking activity has to be somehow detected downstream. So imagine those place cells are sitting here, and they're producing spiking activity. If the spiking activity is not, detected and processed downstream, this information is basically lost. It's non-existent. So, which means, from the point of view of this model, is that every simplex has to basically come with some kind of a readout neuron or readout network, and every single, coactivity combination it requires some kind of readout mechanism. But that is immediately a problem, and the problem of mathematical nature, because you can ask how many of these combinations do you have? And if you make even most basic estimates, you have thousands of cells are active, and number of coactive cells that are coactive at every given moment is, let's say, hundreds. And if you look at the numbers, it's basically binomial coefficient. You have n choose k, n being like thousands, k being like hundreds, and as a result you have little astronomical numbers. So it's obvious that this approach cannot possibly work. Something needs to be done, and what needs to be done is kind of clear. You basically need to say that well, if we cannot count all coactivity combinations, we have to count only special ones. We have to extract combinations of cells that are in some sense more important than others, and call these combinations special combinations. And what I'm saying now is just a logic of what is happening. And what's interesting is that physiologically, this is exactly how things work. Like there are actually special combinations of cells which are called cell assemblies, and the cell assemblies are, well, groups of cells which are frequently being coactive. It's the groups of cells that like each other. They every time are showing coactivity together. And basically the idea is to use, question is this, can we kind of identify these combinations of cells and build a simplicial complex, not from arbitrary coactivities, but only from these special ones. And since the special ones are called cell assemblies, I'm going to call this cell assembly complex. So the key task here is to reduce the number of these combinations. There's other questions like, I would not like them to be too large. I don't want to have too many cells in a combination and so on. But the key idea is how to reduce the number of coactive combinations. And what can be done, we actually did this kind of analysis. And what you are seeing here, these are centers of place cells in this two-dimensional environment that I showed before with one hole. And we basically applied a threshold and selected combinations of cells that are being frequently coactive, most frequently being coactive. And these cells, they get a readout neuron. So what they're seeing here is underneath this layer of neurons underneath, these are readout neurons. And when this happens, we can show that we basically achieve our goal. I cannot go into too many details. I don't have so much time, but it's possible to get number of cell assemblies similar to number of cells. And this equation or this constraint is dictated by anatomy of hippocampus. And we can show that, these cell assemblies, they are nicely overlapping, like a group of cells that participates in one cell assembly and the group of cells that participate in another cell assembly, they overlap by about 80%. So when the rat is running in the environment, this activity of the place cell activity in the hippocampus, it propagates almost, I cannot say continuously, but it's contiguous. You know, it just the activity goes smoothly. Let's say it can be. You know, it's a big topic. What does it mean actually? This smooth or contiguous succession of activity in the hippocampus, but this is what is happening. And most importantly, as you reduce the number of coactive combinations, you actually still able to produce correct topology, and learning times don't change so much. So in the end, what we have is we essentially can implement this whole topological construction in a network. Like before this step, it was basically a phenomenological description of information, and now we can actually reduce it to an actual, you know, neural network construction and show that the whole thing can work. Next step, if we have this neural network construction, then okay, imagine what is happening. We have a bunch of place cells spiking, and they're sending spikes downstream to readout neurons. Now, it's one thing to say that every place cell combination has a readout neuron, but if we consider the physics or if we consider the physiology or physics of this process, what is happening is that neurons are like this thing here is representing a readout neuron, and this readout neuron is getting inputs from place cells. But you can imagine that the way this information is being processed, there are at least two scenarios. In one case, this readout neuron, what it can do is it can spike in response to receiving nearly simultaneous coactivity of cells. So basically you have this readout neuron and it has to receive inputs almost simultaneously, and then it responds. These kinds of neurons are called coactivity detectors, and they actually do exist. But but you can also think of another scenario in which a neuron spikes as a result of integrating spiking activity over an extended period of time. So first, second, and then third, and then it spikes. So the process of collecting inputs is extended over time. And as it turns out, at least these two, they can be more complicated scenarios, of course, but even these two, you can actually show that the effect that they produce is actually quite significant. So let me explain what you are seeing here. So we modeled topological structure of this coactivity pattern, like, this hippocampal activity in a bat. So bat is lying in 3D. And this is what happens if you detect coactivity simultaneously. And this is what happens if you integrate spikes. And you can see that if you try to build your coactivity, if you try to build your coactivity complex by detecting simultaneous spiking of place cells, then this simplicial complex is literally riddled with two-dimensional bubbles. Just it looks like a Swiss cheese, literally, and it has a lot of holes, and they never go away. So it just does not work. But if you integrate spiking activity, if you allow yourself some time, it's a separate question how much time you allow those neurons to integrate. But if you do that, then all of a sudden you have a very finite time. It's slightly large, but it's finite, like something like 15 minutes, and you get correct representation of to the three-dimensional space. So the conclusion would be, and yeah, to make a distinction between these two cases, we call simultaneously detecting readout neurons and simplicial complexes they produce. We call them simplicial complexes as opposed to click complexes. So simplex, this is referring to simplexes, and this is referring to clicks in graph representation of coactivity. But okay, this is a technical side, never mind, just forget this. Just what they are supposed to see here is that if you detect coactivity as opposed to integrate coactivity, huge difference in properties of the resulting map. So the prediction would say that it's if the animal or particular organism is involved in representing complex environments, then the prediction of this theory would be that readout neurons sitting, for example, downstream from hippocampus, in the hippocampal loop or in the cortex, it's better to integrate inputs as opposed to detect. And this is in principle something that can be verified in experiment, how these neurons are processing their spikes. The prediction of the theory is that they should integrate. Now, one more step. Let's say we agree that we're integrating our outputs. So we're using integrating neurons. Still, we can ask more questions. For example, like if these cells, like here are cell assemblies, place cell assemblies, these are readout neurons. When cell assemblies, when place cells fire, they are sending, action potentials downstream to readout neurons. But this action potential is going to be sent, like there are many aspects of the mechanism that how the action potential is being sent. A simple way to describe what is happening is to say that this action potential is traveling downstream with the probability p less than 1. It's a very, very simple, very basic description of effectiveness of synapses. We're trying to introduce properties of synapses into the picture. And another thing is, if you think in terms of this readout neuron, let's say readout neuron received all the inputs it needs, but whether it's going to respond or not, it's another probabilistic process. It may respond and may not, or it will respond with probability Q. So we have at least 2 parameters to play with. You can associate every connection with a probability P and every readout neuron's probability Q and see what happens to that, topological map formation. And this is what happens. Now, this is this graph, you should probably read from, from right to left. When probability of, so this is probability of transmission, this P, and this is learning time. When probability is large, so you are sitting close to 1, you have some, learning time. And again, I'm talking about this one, this little square. The environment is the same. Then when probability goes down for a while, when you go down by almost by 10%, nothing really happens. So, and then when you go even more down, then learning time begins to increase, but then you go, and variability also increases. But then if you go down to some critical value here, all of a sudden you cannot pass through that point. It basically means no matter how much your neurons are trying to spike, your connections are too bad to produce a meaningful topological map. So these graphs are showing similar dependencies for learning time as a function of Q and the combination of those. But basically this graph explains everything. It just shows basically the idea what is happening as the probability of sending information and receiving information is going down. It takes longer and longer to produce a map. So this is dependence of how learning times, they depend as an inverse power law, as a distance, as a function of distance from this critical value. Both for there is a critical value for P and critical value for Q, and this is how things depend. And yeah, I don't have time to talk about this on topological. What's happening is just basically we can describe dynamics of topological defects in the simplicial complex, but the idea is basically the whole idea is here. Now, interestingly, if you look at what is happening in terms of learning region, when probability of transmitting spikes, this is a function of P again. The probability of transmission spike is high, you have a nice, healthier looking learning region. But then the probability goes down, this learning region it becomes smaller and redder because you are actually spending more time to produce a topological map or simplicial complex with required topological properties. But then when you get to a certain point, learning region disappears entirely. So what it means is that if you're, as I said already, is that if your connections, synaptic connections are poor, no matter how your neurons are trying to transmit information, there is no way of doing this. And I mentioned before the case of Alzheimer's disease, they actually observe, and in many neurological diseases, they observe that spiking activity is, as disease is progressing, spiking activity of neurons grows. So neurons are trying to compensate for what is happening, but they simply cannot after a certain point. So this is another way of, topological kind of way of looking at Alzheimer's disease. I'm sorry, it's just. So one more thing I wanted to go over, one more effect. These cell assemblies that I mentioned before, this frequently co-firing combinations of place cells. Now, the problem is that these combinations, they emerge dynamically. this combination of the place cells, they appear as the result of coactivity, but then they can very quickly decay. So what you are seeing here is what I've showed before, like a bunch of cells connected to readout neurons. But in reality, what is happening is that, groups of cells that are forming an assembly, they can kind of redistribute. You have one assembly appearing, then it decays, another assembly appears and decays, and so on. So it's a very dynamical kind of transient network. But that poses a very serious question. How can it possibly be that you are dealing with a dynamic, a transient neural network, and yet you expect to produce a stable topological map? Like the time scale at which cell assemblies are changing is, let's say, seconds, for example, or minutes. It depends. It's very difficult to estimate it reliably, but let's say seconds, tens of seconds. And cognitive maps can be stable for weeks. So how can it possibly be that you have a transient neuronal stratum and stable outcome? Now, what we did was it's actually very easy to model transient network. Basically, you create connections, as I was showing, connections within cell assembly between place cells, or between place cells and readout neurons. You just attribute some kind of a decay constant to connections. So connections, they appear and then disappear. They decay at the rate of, in this case, it's 9 seconds. So you see, this is a typical lifetime of a connection in a network. So what you are seeing here, these are timelines of simplexes. They appear, they last for a while and disappear, appear, disappear, appear, disappear. And the color corresponds to dimensionality of the simplex. You see that bluer simplexes live longer because they're lower dimensional and they live longer. Now, what happens, again, I don't have time to go over all of this, but what happens is if you look at the simplicial complex, it's interesting what is happening. It kind of preserves its size. So if you look at the number of simplexes in the simplicial complex, it more or less stays the same. If you look at the correlation from one time step to another time step, or change over small time period, the correlation actually is very high. Like, I'm not going to go into detail what correlation means, but intuitively it should be clear. If you look at the simplicial complex and compare it to what it's going to be like at the next moment of time, they're similar. But if you look at a similarity over a larger period of time, then the similarity, any similarity measure would come up with, it decays very rapidly. So simplicial complex at the moment T1 has very little in common with simplicial complex like after a minute, for example. However, if you look at topological shape of this complex, look at the Bethe numbers. Again, this is one-dimensional loop, and these guys represent, I believe, two-dimensional and three-dimensional loops we computed. It turns out that in principle, it depends on what is the decay rate of connections and how rapidly things change or how rapidly things rewire. But in all cases, the dynamics of topological loops is much slower than the dynamics of individual simplexes. In other words, your cell assemblies can rewire rapidly, but the net shape of the simplicial complex, the shape of this cognitive map that they represent, changes slowly. And what's interesting is that if you actually increase the rate, it's basically to say that this fluctuation is very easy to interpret. You can imagine that the rat if the rat is thinking that it's running in this kind of environment, then these topological fluctuations, they basically mean that you have like extra loop appearing here and there, extra bubble, or the loop is closing. So this kind of dynamics is happening, topological dynamics. But what's interesting is that if you slightly increase or let's say decrease the rate of decay and bring it, let's say, but still it's very finite. The rate, the connections decay, let's say, over 15 seconds or so, if I remember it correctly. Very short period. It turns out you can completely, despite having these rapidly decaying cell assemblies, relatively rapidly, you completely suppress topological fluctuations in your map entirely, which is kind of interesting, because what is happening is that you can argue that, geometrical shape of the complex is changing, but its topological structure remains stable, even though, I repeat again, that the stratum, the wiring of the network is changing all the time at the rate of, the rate of seconds. But the topological structure is completely 100% stable. So it's like interesting phenomena of separation of time scales, I would say. Like, at the micro level, dynamics is very intense. At the macro level, there is no dynamics at all. The whole thing is very stable. It just freezes out. It's like a, I don't know, interesting physical phenomenon or mathematical, I don't know how to call it. Now, I would like to, if I have time, to mention two things here. Like we've basically applied all these topological ideas or semi-topological ideas to describing different aspects of hippocampal activity. And every time it seems to work, or at least every time it seems to make sense what the model predicts. And the question would be, what kind of implications does it suggest regarding the nature of hippocampal map? What is the big picture here? And if you think about that, basically what we were saying all this time is that spiking information, coactivity between neurons, it represents a pattern of spatial overlaps. And we're basically interpreting these patterns of spatial overlap. We can formalize it in terms of simplicial complex, for example, but it's just a formalization that ultimately spiking coactivity represents pattern of spatial overlaps, containments, adjacencies between regions that are represented by place cells. Interesting thing to notice is that if you are given a particular pattern of overlap here, you can actually build maps of different shapes, right? I give you this part of the left side of the screen, pattern of coactivity. What does it correspond to here? It can be this, it can be this, it can be this, different shapes. So one thing that we can extract from this is that maybe that hippocampus, it's a common phrase that hippocampus represents space. What does it represent about space? What aspect of space does it represent? And from this perspective, it seems like this whole analysis seems to suggest that hippocampus ultimately represents a topological map, meaning it's a map like a subway map, right? As opposed to geometrical topographic map, quantitative map. This map has, it's a map, subway map of Singapore. And this is a map of Singapore. And basically, geometric map contains a lot of information that is not present here. Here, you have a lot of information about distances and angles and scales. And one hypothesis would be that hippocampus actually does not represent all this. It does not represent all this. It represents this only. So this is just an experiment. This is an experiment I've done at UCSF so many years ago. So what you are seeing here, it's a top-down view on one-dimensional track. The rat was running like this, and then it was running back. And as it's running, the track itself is changing its shape right under that rat. Basically, it was running over this changing environment. And the idea was that if this idea of geometric invariance is correct, then spiking activity should be basically, it should not change. And it basically did not. Of course, like one experiment would not prove much. And, I know very well that I'm not the best experimentalist, so my data was not maybe ideally clear. But, prediction of this approach would be that if spiking activity will be recorded in slowly, and it's a keyword, slowly changing environments, morphing environments, the spiking activity would be invariant with respect to those changes. It's like a prediction that I would like to mention. Basically, it's an invitation to, if somebody wants to see how things behave in experiment, that would be my prediction, basically. So upshot is that hippocampus actually represents a topological map of space, not just a map of space, but topological map of space. And I have very little time, right? 5 minutes. I'll try to quickly go over. So when we talk about place selectivity, this defines a discrete set of locations. I would like to ask a semi-philosophical question. What is a location actually? What do we mean by we're saying we're representing a location? And computational location, just an instance of coactivity of so many cells, right? What is a if you think about that? Or you can think about as a portion of information that is received by downstream neurons. That's another way of looking at this. From the cognitive perspective, you know, if a location is some kind of a minimal memory element or memory episode. So we can make a shift here and say that, okay, if we may actually turn things around and say that hippocampus, like its main function is to encode memories. Space is only one of possible like types of memory. So we can say, okay, what if we actually turn table around? Basically, we'll say that we'll think of memory episodes as locations or domains in the memory space. We basically would introduce this concept of memory space, and coactivities of neurons represent locations in that memory space. So from that perspective, when the animal is actually navigating in space and we're observing this, putting our observation aside, where the animal is navigating, it's navigating like as far as its hippocampus is concerned, the animal is navigating in the memory space. Let's look at it that way. So every cell assembly represents, you know, a portion in memory space or location in memory space. We can think of it like this. And group of assemblies represent like memory domains in that space. And that space is represented by a simplicial complex, as we've discussed. I don't want to go into this any, you know, deeper, but and this simplicial complex basically was a schematic representation from that perspective, schematic representation of memory space. And as the rat was navigating, you can see that, you know, the rat goes from point to a schematic representation of what is happening. At the same time, you have this active simplex moving over the simplicial complex and tracing what I'm calling a simplicial trajectory. So movements in space or in memory space are represented by a simplicial, the chain of simplexes, like simplicial path. So at that point, we'll say, okay, the coactivity complex is a schematic model of hippocampal space in which every simplex corresponds to a location. But think about, like, we can look at it as a prototype of a, you know, of a space, if you will, in which every simplex, like, think about, you know, this is a piece of simplicial complex. This is a simplex sitting somewhere here, right? So the simplex actually schematically represents an abstract location in a memory space, and simplexes that surround it, they represent a vicinity. Now we can take this picture and take it one step further and say that, you know, we can translate it into proper topological space, going from, you know, from algebraic topology, so to speak, to general topology, in which with this define spaces, construct spaces explicitly by introducing points and introducing as proper locations, introducing vicinities, topological vicinities. If we do that, again, I have like 2 minutes left. So basically, if we do that, we come up with a constructive model of a topological memory space. And it's a very, you know, it's a well-known beast in topology. It's finite topological spaces, or Alexandrov spaces that are called. Alexandrov was the one who introduced this concept of finitary topological spaces. And in that way, we basically build a constructive model of a memory space. The difference is that this word constructive, because simplicial complexes, they work fine, but basically simplicial complexes simplicial complex is a metaphor for what is happening. It's a way of description, it's a schema. And this object is an actual object. Basically, memory space, if I can say it in these terms, memory space acquires an ontological identity. We're talking about memory space, and here's a space for you that actually like models that space, and it is a space in proper mathematical sense. And this kind of, it's easy to show that topological shape, notice this word shape, in terms of like loops and everything, these shapes are the same. So what we're actually achieving is this accuracy of description or ontological accuracy of description. We're passing from a schema to an actual space, and basically going back to, and you can actually show that when you move along a trajectory in space, you are tracing a continuous, topologically continuous trajectory in that memory space. And this is a simulation of this process, and I have 15 seconds. Basically, what I was, going back to the original slide that I started from, how does brain represent space? One question is, how do you integrate spikes and produce produce geometry? This would take another, 2 hours of talking, but basically at this point, I can argue that this is an example when you integrate spikes and you produce a topological space. So you it's an example of crossing that, of making that link from spikes as complex temporal code to an object of geometrical / topological nature. So we made that link from spikes to spatiality. And that, I would say, is it. I would not spend time on this.
Yuri, thank you very much for this fantastic